Greetings, and welcome to the Quick Start Guide to Transcribing New York Botanical Garden Specimens through Notes from Nature. By watching this video, you'll receive the training needed to participate in virtual citizen science expeditions designed to help unlock the secrets held by historic biodiversity collections. We'll first cover the basics of navigating the platform, Notes from Nature. We'll then break down all the objectives for data capture and explain how to identify crucial information from herbarium specimen labels. Last, we'll introduce all the essential resources you will likely need to consult through the course of your work. Before continuing, feel free to pause this video to learn more about groundbreaking plant science at the New York Botanical Garden and also the growing importance of digitized museum specimens from our amazing collaborators at iDigBio. For 125 years, New York Botanical Garden scientists have led the world in plant research, but your journey is just about to begin, so let's get started. The Notes from Nature virtual transcription platform allows citizen scientists working from anywhere to access natural history specimens from museum collections and provides the tools needed to record and report essential data about them to experts in the field. Direct links to all the latest NYBG expeditions on Notes from Nature can be found at the Linda Steer Herbarium virtual volunteer page. The first time you open an expedition, the project tutorial will automatically appear. Scroll up and down to see each page fully, and click Continue to proceed from one slide to the next. Try to read the full tutorial before completing your first transcription. You can always exit out by clicking the X at the top. You are now stationed at the primary interface for transcribing New York Botanical Garden specimens through Notes from Nature. We recommend that all new users register for a Notes from Nature account, then make sure to sign in whenever working with the portal. Doing so allows access to important features, such as leaving comments and tracking your contribution history, which you can always check by clicking the Field Book button. The section of your screen on the left is the specimen display, where you will view images of historic preserved plants from the William and Linda Steer Herbarium. On your right is the data entry window. Here you will enter scientifically relevant details about the conditions of collection for each historic plant specimen. In the middle column are display navigation tools. Use the Pan tool to search each historic specimen for its primary collection label. Click Zoom in to enlarge the image magnification until the text on the label becomes easily legible, and use the Pan tool again to center. When the Pan tool is activated, the mouse wheel can also be used to zoom in and out on the specimen display. The Notes from Nature interface divides your transcription objectives into several pages. Once the fields on any given page are completed, you can click Next to navigate to the next page. If you need to return to an earlier page, you can click the Back button within the Notes from Nature interface. Be careful not to mistakenly click the Back button in your browser, since that will erase all of the work on the current transcription. If you need assistance with the fields on any given page, click the nearest Need More Help with This Task button to consult the help text for that task. After completing the final page, if you are satisfied with your transcription, click the Done button to finalize and submit your answers. If there are comments you need to make, in particular regarding difficulties encountered with the current transcription, you can click the Done and Talk button. This automatically activates a new forum module called Talk within Notes from Nature and allows you to type a textual note that will directly accompany your submission. This comment will be visible to all other virtual transcribers and the project administrators. For a complete set of standards and guidelines for writing clear and operative talk comments, visit the NYBG Expeditions forum post linked here. Now we are going to do a quick run-through of all the transcription objectives for NYBG Expeditions and outline the basics for how to identify target information from herbarium specimens and then enter data into the appropriate fields. On the first page, we see 3 fields relating to higher geography. Here you must determine the county, state, and country for where on Earth this plant was originally collected. These parcels of information should be clearly indicated for each specimen, usually at or near the top of the primary collection label. Hierarchical drop-down menus are provided so that each selection affects the options listed in the next field. If the country of collection isn't shown, it should be inferred from state. Otherwise, if a value for any of these three fields is absent, select the answer not shown. The field titled Location asks for a specific description of the place where this plant was collected. This can include nearby towns, cities, or other destination names, description of proximity to rivers, lakes, or road intersections, and any other manner of geographically relevant directions. Your goal is to type into this one text box all written information that seems useful for locating the site, as close as you can to how it appears on the label. You should avoid re-entering geographic information called for by other dedicated fields such as county, elevation, or latitude and longitude. Include descriptions of habitat features only if they seem obviously useful for locating the place of collection on a map with greater accuracy. If the altitude of the place of collection is shown on the label, use this dedicated field for entering that numeric value in either meters or feet. If presented as a range, use this field for entering the lower value and this field for entering the higher value. Use this drop-down field to specify the units whenever an elevation value is entered into any field. On the next page, dedicated fields are provided for entering latitude and longitude coordinates that may appear on specimen labels. These coordinates, which quite precisely identify the location of collection, are very useful for scientists looking to model the geographic distributions of plant species. Be sure to follow the detailed instructions for entering latitude and longitude given in the Need More Help section on this page. Primary collector is always the lead scientist who collected the plant specimen in the wild. Their name should appear prominently on the label, usually near the top or the bottom. It is important to enter last name first, followed by a comma, then first and middle names or initials as shown on the label. Identifying the correct primary collector can be difficult for some labels, but becomes much easier with practice, as you learn to identify patterns and differentiate between other sorts of names, such as determiners, which also appear on herbarium specimens. Make sure to consult the help text for this field and watch several examples of complete transcriptions before you get started. Suggestions for handling common situations are given in our Expedition FAQ guide. Other collectors asks us whether any colleagues or collaborators accompany the primary collector on this expedition. We enter the names of all other individuals listed after the primary collector's name verbatim as they appear on the collection label without switching the first and last names. Collector number is a unique sequential number assigned to every specimen a botanist collects over their lifetime. Used in conjunction with the primary collector's name, this number is an essential mechanism for tracking herbarium specimens as they are used for research. Last but not least, each specimen has a collection date. Here we are asking for the year, month, and day that this plant was found in the wild, which forever establishes this specimen's place in the historical record. Simple dates can be entered using only the first three fields on this page. If, however, multiple dates spanning days, months, or years are given on the label, use start date fields to specify the earliest date and end date fields to specify the latest date. By now, you should have a basic understanding of all the units of information targeted for capture from herbarium specimens. Identifying these data on collection labels will grow easier as you become more familiar with various label formats. In difficult situations, the most important thing to know is where to look for help. First, remember to always check the help text and reread the project tutorial. Your next stop should be the NYBG Expeditions Help Forum, where you'll find numerous posts dedicated to providing solutions to a wide array of common problems. After signing in to Notes from Nature, you can click subscribe to receive an email update whenever new guidelines and instructions are posted to this page. You'll come to learn that in many situations, the best tool can be the very database we are looking to build, the CV-STAR Virtual Herbarium. From this page, you can obtain higher resolution images of any NYBG specimen viewed in Notes from Nature. You can also find clues that help you interpret information from herbarium labels by searching for related specimens which share a collector, date, and/or location and have already been fully transcribed. Inevitably, you will encounter some specimens that can't be understood, either due to illegible handwriting or misleading or ambiguous information. Submitting your answers with the Done in Talk comment is precisely the correct mechanism for coping with these situations. Always make sure to add the correct tags to your comment so your issue can be interpreted quickly and correctly. This concludes the Quick Start Guide for transcribing historic New York Botanical Garden specimens using notes from nature. See other posted videos, including complete transcription examples, and find other resources available for citizen scientists at the William and Linda Steer Herbarium volunteer page.